You guys still doing all right? Everything coming out Yeah, I'll go bye-bye.
You going bye-bye?
I go bye-bye. Yes, I will go bye-bye with you.
People think it needs more light being brown. And if it's more light, you bleach it.
Yeah, but it's
the Community Institute of Ocean Sciences. We did a course under coral reef ecology. The course included laboratories and field techniques, including scuba diving. Some of the stuff we did specifically while diving was video transecting of reefs. So we'd go down and we would lay down transect lines that were 10 meters long. And we took a camera with us with a 2-foot pole on the camera, and we dive right over the reef. And you have to keep the camera as close to the reef as you can without hitting it. And then we dive along it. And then we took the video clips and we take them back into the laboratory and just put them up on a computer screen. And we basically just put a clear sheet of paper over the computer screen and put 10 random dots, 10 random black dots, 10 random red dots. And we would play the videotape through, and every 7 to 10 seconds we would stop it, and we would, whatever the dots landed on, we would quantify it. If it landed on like a brain coral, if it landed on fleshy algae or sand or rock, we would mark it. And it allowed us to assess the actual community of the reefs down there. So that was one thing that we actually used. Another thing we did was quantifications of the reef fish, the makeup of the community, and pretty much the same thing, pretty simple. We laid down a transect line, just swam along it, and with a clipboard just took notes and bring it back to the lab. And what we did was we did it on the outskirts of the reef, we did it in the middle of the reef, and then we did it on the land side of the reef. And we wanted to see which functional groups of algae were growing where and how they were affecting the actual corals that were growing. So we collected them, bring them back to the lab. We separate them into functional groups, like some are Peltarians, some are Filamentous. So we break them up like that, not by species, just by functional groups. And then we bake them in the oven to get a dry weight of them. And we use that to quantify the actual biomass of the algae that were growing. What else did we do? We did coral IDing, which is pretty self-explanatory. We did a coral bleaching assessment. So we had to learn about a few of the major types of diseases that are occurring down there, and then they took us to a site that they knew there was going to be, they knew was affected, so that we could actually gather some data. And we went down and we had to generally measure the full size of the coral, and then measure out the percentage of the coral that was actually bleached out to the point where it was just open calcium carbonate. And so we did that, and we also did another survey for juvenile corals. In the 60s, I believe it was, there was a big ship. So Bermuda is an atoll, so it's an island that is kept up by the coral reefs. It was an island that's been washed away by the ocean, but the corals is the only thing that's keeping it up and keeping it an island. So it's pretty treacherous to actually get to the island if you don't know how to navigate. And in the 60s, a big boat wrecked into a reef. And ever since then, BIOS has been going back every year to measure the juvenile corals, to see which corals are actually making up the next generation of the reef, and monitoring how fast they're growing. So that was one thing we did, was we went out there and with calipers we went down there, measured, we laid down a transect line. It was pretty key to have transect lines 4 to 5 on us every dive. So then we laid down the quadrant and we measure all the break them up by species, the corals, and we measure them and bring them into the lab to get a general idea of the area of new growth of coral compared to past years of the class. We try to do an experiment for the invasive species of lionfish, which is pretty invasive pretty much everywhere nowadays. When we actually went to do them, they won't let us spear them. We actually had to take a class to learn how to spear them. Normally, if we were out of we'd be able to, but because we were with them, they wouldn't let us. And when we went to actually find them, I think we only found about 3, so it was pretty unsuccessful. But we later did a deep dive, like 130 feet, and I think we came up with like 8 or 9. And the one reason the lionfish are so horrible to the reefs out there is really there's no predators for them. They would like to try to introduce something that could take them out, but they have nothing so far. And they also can eat a tremendous amount, so they're just destroying small reef fish in large quantities at a time. And then, so we would all go and present, and so on one dive I would go and collect information on the fish structure, and then the next dive I would go and collect algae samples. And there was 8 of us in the class, so we broke up into 4 groups. And then at the end of the 3 weeks, we would all be entering in our own data into the computer. At the end of the 3 weeks, we got to work with a partner and pick one major project that we worked on. And the project that me and my partner worked on was the comparison of zooxanthellae density and the chlorophyll concentration in two distinct sites in Bermuda. So because it's an island, they try to burn off as much garbage that they can if possible. It's more cost effective that way. So they have this plant where they just burn garbage, and it's right on the coast. And what they do is they suck in water from the ocean and they use it as coolant, and then it pumps back out to the ocean. And they claim it has no leaching out, which we didn't test for in any way. But either way, the water returns back out to the ocean about 10 degrees warmer than actual seawater, which was already pretty warm. It was already about 78 degrees. So we collected samples of Madrasis aurotenra from 2 sites, one from right outside of the outlet of the pipe, and then another site which was farther away and it was supposed to be a more pristine location just to see if we could find a difference in them. And the general experiment was we brought these little frags back, and then we used just a regular bathroom toothpick that you would use in your mouth, and we filled it with salt water, and you could spray off all the tissue from the coral. And then once collecting that, you run through a centrifuge, and all the actual zooxanthellae collect at the bottom of it, and we collected those out. And then we put them through a microscope, and it's called a hemoscopometer. It's basically, it allows you to use a microscope for area. So it looked like a big sheet of graph paper, and then you just counted as many zooxanthellae as you could find in the graph in each square. And which gave us an estimate of the amount of density of zooxanthellae in each individual frag. And after we collected that data, we put all of the zooxanthellae through acetone overnight. And putting in acetone extracts the chlorophyll out of the zooxanthellae. And then we ran it through a spectrometer, which reads absorbance. We shoot a light through it, it reads absorbance wavelength back. And that was to determine the chlorophyll concentration. And so basically, what we found, none of our data ended up being significant. The class size that they can normally have is 16, so normally there's twice as much data as we collected. So that was one thing that could have messed with our results because we just didn't get enough data. But what we found was that there was less zooxanthellae actually found at the corals that were by the outlet type, which was what we expected. So it seemed as if the corals were stressed out and they had expelled some of their zooxanthellae. And the corals that were in the prestige area had more zooxanthellae. But when we did the chlorophyll concentration, we realized that the corals that were actually at the stressed environment actually had more chlorophyll in them than the corals in the prestige area, which was intriguing. But what we assume but can't conclude on is that the corals outside of the outlet pipe are under more pressure, more environmental pressures, and so that they possibly the plant's been there, I think, for at least 60 years. So they've had some time to adjust to the actual change in temperature a little bit. So what we assumed was that they've had some time to adjust, and they've expelled the zooxanthellae that aren't productive and have kept in the clade, the zooxanthellae clades that are more heat tolerant and more productive to make energy. So they actually had less zooxanthellae, but the zooxanthellae they had were more productive overall, which I think allowed them to balance out and still survive. I did want to bring in information. I thought that was my experiment that we worked on. I did want to bring in the information for the herbivory on the reefs, but I tried to get a hold of the girl, and she's in the UK, and she hasn't gotten back to me yet. I tried to get her report, so it's all of our data, but they ended up doing a report on the herbivory. But I can go into a little bit about it. So we wanted to look at the overview of Bermuda reefs, which were generally amazing. Like even when they took us to a bad site, it still looked absolutely gorgeous. So we were looking at good sites and bad sites in Bermuda, and then we wanted to look at good sites and bad sites to other reefs in the world. And so Jamaica was chosen because they've been going through a lot of problems with Jamaican reefs. Part of it is due to overfishing. Part of it is due to, I think in the 70s, they had a huge die-off of urchins. I think it was coupled with 2 major hurricanes. And so basically there was nothing to control their algae. And that coupled with killing off the herbivorous fish due just to lack of food for people has caused their reefs to just be completely degraded and completely algae covered. And so we were trying to make assumptions and connections between the Bermuda reefs and Jamaican reefs, but I wish I would have had more. I wish I would have been able to get their actual document on it, all the data. I only had mine, so. But yeah, I mean that was pretty much the general setup of it. Like I said, the class was 3 weeks long. At the end, we had a couple tests during it, and then at the end, we just had to present a speech like this. Numbers have been like decimated, and we're a little worried that it might not actually bounce back.
We've recently seen 2 stores get plead guilty for pulling coral on protected reefs. How much through your studies have you seen much damage to coral being going out?
Not there, because honestly, the island's pretty small. It's like 3 square miles. So on the island, I don't think I really saw any like aquarium like stores. So I mean, I guarantee it happens. I mean, there's no way about it. But they do have protected areas, and they've been working with the fishermen, not so much specifically with the coral, but at least with the fish. And they've been the fishermen are actually finding the benefit of having protected areas. They live in those areas, and then as they start to grow in population, they obviously just spread outwards. And so they'll literally go right to the line, and with a GPS, they'll know exactly where they can go, and they'll fish right on the line of where they can. And they're actually finding the benefits of having protected areas because their actual bring in for their energy is
So you think it's more protected for the fish than it is the coral?
Yeah, it definitely is.
Okay.
It's definitely what their main focus is on right now, at least with their conservation laws. I think it's easier for them to control because they require the fishermen to document what they're bringing in and how long they're fishing. So it's easier for them to quantify how much is actually being taken out. So yeah.
Anybody else have any questions for him?
How fast do the stony corals grow?
How fast do they grow? Like we only took measurements for this specific year. We didn't actually get to use the data from previous years, but it's actually like, so the research center, it's not really like a school. It's a research center that has an education background. So there's people there that are working year round. So it's almost like we're working for them. We go out there and collect their data, and then they're going to eventually, every 10 years or so, republish it into a newspaper and stuff like that. So that's more the aspect of that. At least reflecting that data more for them for a long period scale. We got anything else? Let's see. I've had to give previous reports on this, so I just grabbed an old paper, but most of it's more about explaining the program that I actually did there to younger students, just to show them the research opportunities that are out there.
We got a guy right here that would like to.
Travis is talking about taking marine biology.
So if you get a chance before you leave, have a talk with him.
Yeah, definitely. I'm so glad I did. I spent 2 years at community college and I wasn't going to, and then my girlfriend at the time